This brief presentation will explore how PEA interacts with the human endocannabinoid system and how it produces anti-inflammatory and analgesic benefits. In the early 1960s, researchers isolated THC and CBD, the primary cannabinoids in cannabis. Because they are plant-derived, these exogenous cannabinoids are sometimes called phytocannabinoids. Nearly 30 years later, two endogenous cannabinoids were discovered, anandamide, or AEA, in 1992, and 2-AG in 1995. These endocannabinoids are produced on demand in human tissue. The anti-inflammatory and analgesic benefits of both exogenous and endogenous cannabinoids occur when they interact with specialized receptors in our bodies. There are four receptors that we should understand. CB1 receptors are found on presynaptic neurons in the brain and central nervous system. When they are activated by either anandamide or 2-AG, they downregulate neurotransmitter release. Although CB1 receptors are responsible for the psychoactive effects of THC, they are not directly involved in reducing pain or inflammation. The next three receptors are CB2 receptors are found in the tissues and cells of the immune system, including the thymus, tonsils, and spleen, as well as macrophages, lymphocytes, and monocytes. While CB1 receptors modulate neural activity, CB2 receptors modulate immune responses. When inflammation occurs in our tissues, the endocannabinoids anandamide and 2-AG are produced locally and released into the area. They bind to CB2 receptors on nearby immune system cells, reducing their activity. Activation of CB2 receptors therefore produces a natural anti-inflammatory effect. Much like CB2, PPAR-alpha receptors are also primarily found on immune cells, particularly on mast cells, which normally release inflammatory cytokines and chemokines. Anandamide, 2-AG, and other endocannabinoids, which are synthesized on demand in response to local inflammation, bind to and activate those PPAR-alpha receptors, reducing mast cell activity. This produces a natural anti-inflammatory effect. TRPV1 receptors are located on the cell membrane of peripheral sensory nerves. Activation of the TRPV1 receptor initiates the transmission of pain. However, certain cannabinoids will actually desensitize the TRPV1 receptor, leading to reduced pain sensation. Let's quickly review these receptors. CB1 receptors are in the CNS and aren't directly involved in reducing pain or inflammation. CB2 and PPAR-alpha receptors are found on immune system cells, and activation of those receptors will reduce inflammation. And finally, TRPV1 receptors are responsible for the transmission of pain signals in peripheral nerves. Let's look at how the two primary endogenous cannabinoids, anandamide and 2-AG, interact with these receptors. Anandamide binds strongly to the CB1 receptor, but only weakly to CB2. It also binds weakly to PPAR-alpha and TRPV1. 2-AG binds strongly to CB1 and CB2 and desensitizes TRPV1, but only binds weakly to PPAR-alpha. What about CBD, which is believed to have anti-inflammatory and analgesic benefits? Well, CBD doesn't bind directly to either CB1 or CB2. It binds weakly to PPAR-alpha, but strongly desensitizes TRPV1. CBD also inhibits the breakdown of 2-AG, thus increasing its tissue levels, which indirectly increases the anti-inflammatory effects of CB2. So, what is PEA and how does it work? PEA, or palmitoyl ethanolamide, is an analog of anandamide. They are both fatty acid amides. And just like anandamide, it is produced in our tissues in response to local inflammation and interacts with cannabinoid receptors. Thus, PEA can be considered an endocannabinoid. However, PEA was first discovered more than 60 years ago as a component of certain foods. It can be extracted from eggs, peanuts, and soy. This fact, that it is both produced endogenously in our tissues, but also found in certain food products, makes PEA unique. It is both an exogenous and endogenous cannabinoid. It has a long track record of safety, having been used as an anti-inflammatory and analgesic in Europe since the 1970s, but its actual mechanism of action was only discovered recently. PEA does not interact directly with either CB1 or CB2. Its primary receptor is PPAR-alpha, so it downregulates mast cell activity and reduces inflammation. It also strongly desensitizes TRPV1, reducing pain. And just like CBD, it inhibits the breakdown of anandamide and 2-AG, leading to indirect stimulation of CB2, which further reduces immune system overactivity. This combination of receptor activation explains PEA's clinically proven anti-inflammatory and analgesic effects. Palmitoyl ethanolamide, PEA, is truly unique. It is the only cannabinoid that is endogenously produced in our tissues as a natural anti-inflammatory, yet is also present in certain common foods. Although it is not found in cannabis, it activates many of the same receptors as CBD. It has been proven safe and effective in numerous clinical trials and is now available here in the United States.